Hello student, welcome to the another lecture of biological classification. Today we are going to see kingdom fungi after completion of kingdom protista. Okay, so let's begin. So first thing we go is the introduction of the kingdom fungi. In that we can say it is a multicellular filamentous eukaryotes. Okay, so again we have started with the prokaryotic unicellular, that is bacteria monera. Then we started with unicellular eukaryote, very important kingdom, that is protista. And finally we have landed our first multicellular kingdom, that is fungi. It is called as unique kingdom of heterotrophs. Let's see why it is called as so unique, how it is so unique. Okay, so first exception we have here. Exceptions are important, that's because that's where the questions are created. So when you say a rule multicellular filamentous eukaryotes, there is an exception, yeast, which is unicellular and which is responsible for most of the fermentation, that is bread, beer and wine and all other kind of things. So yeast has been very important to the dawn of the civilization, from food to fermentation to uh, alcohol. Okay, so let's go with the fungi. And fungi, they show great diversity. Fungi, they show great diversity in morphology as well as habitat. Morphology, external character; habitat, the area they reside. Okay, so let's go with the morphology first, then we will come back to habitat. So morphology, they have filamentous multicellular bodies, we have seen, and they have thread-like structures. These thread-like structure are long and slender, and they are called as hyphae. And when these many hyphae come together, they form a network. This network of hyphae is called as mycelium. Pay attention to the terminology, it is very easy. Thread-like structure, it is going to be called as hyphae, and this hyphae's network is called as mycelium. Okay, then we have here habitat. Then we have here habitat. Habitat is the area they reside. So where they reside and what kind of environment they prefer. So air, water, soil, on and in, on animals and plants, and also in, because some are parasites, endoparasites such as candida, but mostly fungi are ectoparasite. They appear on surface or skin. They prefer warm and humid places. They prefer what? Warm and humid places. So warm and humid places are preferred by them. So wherever there is a humidity and warmth, the fungi grows. Next, I think one point is here about their cell wall, and this is a important point. They will ask questions based on this. Pay attention here. The cell wall is made up of chitin, or you can spell it as it is, chitin. Okay, so there is no shame in calling it chitin rather than chitin, as the English is the most phonetically wrong language. What is the phonetically wrong language? When you write and do not speak as it is called as what? Phonetically wrong. And Indian languages are phonetically correct. You pronounce as you write them. So you can call it chitin for spelling purposes or not. It's up to you. And polysaccharides. This chitin plus polysaccharide, this data is from NCERT, so no harm in remembering it. And directly you can use it for NEET questions. So it is made up of NAG, a homopolymer. Okay, chitin is a homopolymer of NAG. What is a homopolymer? Same building blocks. If you use to make a chain or a polymer, it is called as what? Homopolymer. If you use different individual monomers, it is called as heteropolymer. So what is a NAG also? So it is N-acetyl glucosamine. This N-acetyl glucosamine is a modified sugar, okay, of glucose. So this NAG is present where? In chitin as a repetitive units. Remember this. This concepts of homopolymer, what is NAG, what is NAM, and what is glucose, and how their structures are related. This will be covered in biomolecules unit of cell. Just now, remember this. If someone asks you question based on this, you can write that it is a homopolymer of NAG. So this is a mycelium diagram. Many individual, what is there? Hyphae is there, and the hyphae network is nothing but mycelium. Okay, so it's pretty straightforward and simple. We are moving to next slide. So we have hyphae and there are two types. Okay, so one type is this. This is called as without septum. There is no barrier in this. This is called as without septum, with no barrier in this. And because of this, this becomes multinucleated. Because of this has become what? Multinucleated. Because no wall is there. Barrier is cross barrier wall. Septum is not there. So it has become multinucleated, and multinucleated hyphae without any cross wall, it's called as sinocytic. It's called as what? Sinocytic. And because there is no barrier is called as septum. Septum is called as barrier or cross wall. Cross wall is absent, so it is called as aseptate. So it is called as aseptate. Very easy, nothing to confuse. Okay, so this is a continuous tube filled with multicellular cytoplasm, so it is called as sinocytic. If there were to present some septum, it will be called as septate, but it is not. So remember this. And again, these are present in a particular group or class of fungi, that those are called as phycomycetes. Remember, aseptate multinucleated cytoplasm is present in phycomycetes. Next. Then we have septate. So septate have septa or septate and cross walls in their hyphae. Okay. And these cross walls are formed during nuclear division. As soon as the nucleus gets separated, there is a generation. So if this is a cell, pay attention. So nuclear division happens. As soon as the nuclear division happens, two nuclei will be there. In between those two nuclei, what will happen? Two nuclei will be formed, and in between those, this hyphae will be formed. Okay, so not hyphae, septum. Next, so septum creates uninucleated uh, hyphae. There are two types of septum, and those are particular. Septal pore septum, this one is septal pore septum. It is present in ascomycetes. These are the groups of fungi we are going to see. Okay, then we have dollipore septum. Remember dollipore, it looks like a doll. By what way it looks like a doll, I don't know, but it is called as dollipore septum. So it is present in basidiomycetes. It is present in basidiomycetes. So we have gotten here sinocytic hyphae, aseptate hyphae, and septate hyphae. I think this is clear, very simple. If there is a septum, it is septate. If there is not a septum, it is aseptate. Septa formation creates uninucleated cell. Without septa, it will be multinucleated. Very simple. Let's move on to next. So when we are moving on to next, that is nutrition. And we previously known that it is a unique kingdom of heterotrophs. Okay, so all are heterotrophs. Here little bit controversial word framing is there, that most fungi are, they are saying heterotrophic. Most fungi are not heterotrophic. All fungi are heterotrophic. If you depend, what is the heterotrophy? If you depend on other for food, you are heterotroph. Whether you are eating dead or decaying, that is heterotrophic, or you are a parasite living on others, that is also heterotrophy, or you are a symbiotic, that is also heterotrophy, because you depend on other for food. We are also heterotroph. We depend on others for our food. Okay. So most fungi, but pay attention. Whatever NCRT says, mostly right. So follow. Most fungi is correct. So heterotrophic. So first we go with the saprotrophic. Okay. What is this? Saprophyte. So saprophytes absorb soluble organic matter from dead substrate. Whenever the dead and decaying sapro will come, this is just the dead and decaying. They will absorb the content from dead substrate. Second one is parasitic. So parasitic. So parasitic depend on living. Parasitic depend on living and plants and animals, and they will take nutrition from them. Okay. So this interaction again, who is benefited? Parasite is benefited. Who is harmed? Host is harmed. Then we are going with the symbiotic. When we are teaching with the symbiotic, we're going with the symbiotic. Here two symbiotes we have. This is a algae. Okay, so this algae plus fungi becomes lichen. You will study about this at the end of this chapter. There is a special topic called as lichen, and they are symbiotic collaboration with higher roots plants that is called as mycorrhiza. Okay, so if in the future when we are going to study the plant kingdom, there you can see that conifers, their roots are coated with mycorrhiza. They help them survive and absorption. So this is a mycorrhiza in which both higher plant and this fungi will be benefited. Both are benefited. Both are benefited is mutualism. Okay. So here they are calling it symbiotic. More correct term is mutualism. Plus interaction, that is a plus interaction. Okay, so hope you understood the nutrition part is easy. So all our most fungi are heterotropic. Pay attention here, some are saprotrophs, dead and decaying, some are parasites, and some are symbiotic. You have two examples, that is lichen and mycorrhiza. Okay, next, the very important part now, most important part of this is the reproduction of fungi and the kind of spores they produce and their way, whether they are produced inside endogenous or outside exogenous. These questions are asked. This one is important. Okay, this one is important. They will ask you question based on this. So first come here. So in reproduction, spores are produced in fruiting bodies. Spores are produced in fruiting bodies. These are called as fruiting bodies. Something. Okay. So there are different fruiting bodies for asexual reproduction, sexual reproduction, and the four classes of fungi. So specialized fruiting bodies. These fruiting bodies are nothing but specialized mycelium to produce and release spore. We have seen these fruiting bodies in the lecture of slime mold. Okay, we have seen they form fruiting bodies and they release spores. Okay, so let's go one by one. One is vegetative. So vegetative is simply fragmentation, fission, and budding. Okay, so budding example of yeast is very famous. Then what is the difference between fragmentation and fission? Where what is happening? Breakdown is going to happen. There is a mechanical fragmentation. It breaks one into two. So what is the difference between fragmentation and fission? That is just it. It is an accident. Fragmentation is an accident external, and fission is just a division. Fission is the division. This is just a consent. If you are breaking it with consent, it is called as fission. Without consent, it is called as fragmentation. Okay, so accidental, non-accidental, planned. Everywhere this will work everywhere. Don't worry. Then we have asexual mitospore. Then we have what? Asexual mitospore. This asexual mitospore is produced by mitosis, because I have written here asexual spores are mitospore. Mitospores are produced by mitosis. Two types of division, meiosis and mitosis. Meiosis produces sexual spore and mitosis produces asexual spore. So pay attention here. First spore we are discussing is zoospore. First spore we are discussing here is zoospore. These kind of spores are found in phycomycetes. Don't worry, we are going to encounter these classes in detail, but just remember, but just for sake of this, zoospore. And these spores actually move. These spores actually move, and these zoospores are produced within a structure, that is called a zoosporangium. Okay, and they are produced inside. They are produced inside. When the spores are produced inside, it is called as endogenous spore. It is called as what? Endogenous spore. Endogenous spore. Okay. Then we have another spore that is called as sporangiospore. Okay, again same. Spores are produced inside. So again it is called as endogenous spore. So remember, zoospore or sporangiospore, both are endogenous spore, and these are asexual spores. Next, we are going here, that is conidia. So conidia are produced, pay attention, conidia are produced here externally, without any covering, without inside. It is not being produced inside anything, it is produced exogenously. So this is exogenous. Conidia are exogenous. Remember, conidia, whether they will be asexual, whether they will be in a whatever group, they will be exogenously produced. This is simple mechanism. This is exogenous. The production is outside, not inside. Out, exogenous, in, endogenous. Okay. Next. Next is sexual spore, and these sexual spores are produced with meiosis. Okay, so these are called as meiospore. These are called as meiospore. So pay attention here. It is very simple. Oospore. What is there? Oospore. Oospores are haploid. Oospores are haploid. Okay, so this oospore or any mitospore, all spores are haploid. These oospores are seen in oomycetes. So you may ask, where is this oomycetes? This oomycetes and zygomycetes are subgroups of phycomycetes. So this is a sexual spore. Okay, next. Next is ascospore. So it is pretty simple. It belongs to what? Ascomycetes. Again, the spores are inside the ascus. So again, endogenous. And finally, we have basidiospores. Basidiospores are produced like this. Okay, basidiospores are produced like this on the tip. If on the tip it is produced, so on in the basidium it is going to be exogenous, such as conidia. Conidia is asexual spore, exogenous on the tip. Basidiospores are the same. So there is no such rule that asexual spores should be endo or exogenous. Don't make such connections. Very simple. They are different. If they are placed, it has nothing to do with sexual and asexual. If they are produced on the tip, they are exogenous. If they are produced inside some covering, so it is endogenous. It is a high weightage topic. They will ask you question based on this, so pay attention. Now we will consider all these again to remember. So which ones are endogenous? Zoospore, sporangiospore, oospore and ascospore. Which ones are exogenous? Conidia and basidiospore. So there is one terminology here, aplanospore. Aplanospores are nothing but non-motile. Except for all these spore, except for this zoospore, all are non-motile. All are non-motile. These zoospores are present generally in algae, chlamydomonas and all that. So this is present in phycomycetes, which are also called as fungal. of algal fungi. Okay, so we will see about that later. But for now, you must understand which ones are endo and which ones are exo. Okay, next this is zygospore. This is zygospore. Zygospores are diploid, but this is not a spore because it is a zygote. Because it is a zygote, but it is covered like that, and whenever the favorable condition comes, it can germinate. So it is also called a zygospore. So neither it is a sexual spore, neither it's a asexual spore. It is a zygote which is covered by covering. Okay. So this was our reproduction. Next. So I think this is clear. We should move to next topic. So this one is very important. That is called as its sexual cycle. So sexual cycle, how the sexual cycle works is fusion of two protoplasm between two motile or non-motile gametes. When the fusion of protoplasm, they are saying it is not actually protoplasm, but it is cytoplasm. As protoplasm is nucleus plus cytoplasm. But okay, so remember, cytoplasm is fusing, nucleus is not fusing. So cytoplasm fuses first, then nuclei fuse, that is called as karyogamy, and then meiosis happens. So meiosis happens in zygote. Haploid spores are produced. Okay, so if zygote is undergoing meiosis, it is also called as zygotic meiosis. So this is happening. So plasmogamy, karyogamy, and meiosis in zygote. These things are happening. So this is the sexual cycle. Now we are going to see the detailed cycle of this. So we should first start with the two haploid hyphae of compatible mating type. What is meant by compatible mating type? Okay, so the hyphae should be of same species, first condition, and it's better if they are of different strain, same species but different individuals, so that when the sexual reproduction happens, what should increase? Variation should increase. Okay, so we are here. Okay, so this one is plus, this one is minus, whatever you can say. Two compatible type hyphae, they come together and fuse. They form like this, come together and fuse. Okay, so after fusion, what happens is there is one cell which contains two nucleus. These two nucleus are separate. This condition is called as N plus N dikaryotic stage. This condition is called as N plus N dikaryotic stage or dikaryotic phase, whatever you want to call. This is N plus N. This is not 2N. This is not 2N. Okay, that is diploid. This is two nuclei. That's why it is called as dikaryotic. Okay. So when this dikaryotic phase is there, it depends how much time it will take. In some fungi, you can see it happens immediately. So immediately this N plus N fuses, N plus N fuses and makes it 2N. That happens in Phycomycetes. That happens in Phycomycetes. So I remember Phycomycetes has cynocetic hyphae and their dikaryotic phase is very short-lived or immediately they fuse. Next, dikaryotic phase where it is extended, where it is too much longer, it is in Ascomycetes, pay attention here, and Basidiomycetes. Okay, here dikaryotic phase is longer. Also, we know about those both Asco and Basidio that they are having septed branched mycelium. Okay, there is an intervening stage. This stage is also called as intervening. Remember the wording. Okay, wording is very important. Intervening stage, delayed stage, dikaryotic phase. So two nuclei per condition. This condition is known as dikaryon, and the phase is known as dikaryophase. More that, yes. Okay, so after time, after time, they become fuse. After time, after some time, they fuse. Okay, 2n plus 1, they become diploid, fuse, and become diploid. So it takes a longer time taking our usco and basidiomycetes, immediately fusing R phycomycetes. Okay. What happens after they fuse? They form a zygospore or diploid cell. This diploid cell undergoes meiosis. Meiosis part 1, meiosis part 2. It produces four different types of hyphae which are haploid and which contain sexual variation. Variation first due to fusion of different mating types, compatible mating types hyphae, and second due to meiosis, and meiosis generate variation. After that, again one of those hyphae grows up and forms bodies. One of those bodies forms this sporangiosspore, and in this sporangiosspore mitosis happens and asexual spores are produced. These spores are also called as mitospore because they are produced by mitosis, and again these spores give rise to hyphae, and the hyphae again fuse and all that. So it follows the plasmogamy, karyogamy, meiosis again, and this is called as sexual cycle in fungi. Hope you have understood. It is pretty simple if you follow the sequence and concepts. So this is it. Next time we will see the four groups of yes. Next time we will see the four groups of fungi, that is phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes. These are all formed due to the on the basis of morphology of mycelium. Just we have seen septate, aseptate. Okay, mode of spore formation. Just we have seen exo, endo, and the fruiting bodies. Just we have seen the fruiting bodies also, basidium and ascocarp, sporangiospore, conidia, and all this fruiting body, mode of spore formation, and morphology of mycelium. In next lecture, we are going to discuss the phycomycetes, ascomycetes, basidiomycetes, and deuteromycetes. Thank you for listening patiently. This was a very good lecture. I felt very good delivering this to you. Hope you have understood well. And next time we will start with the fungi and their four groups. Okay. Thank you. Bye bye.